Hello, Thibault. We're about to start. Welcome to this third edition of our webinar. Today, we're about to show you how to do omics statistical analysis in Constellab. As last time, at the end, we'll have a short question session. It's a 40 minutes webinar. So we will end by 1 p.m. You can go, Thibault.
Okay. Hello, everyone. So we're gonna to start. So I'm going to share my screen. Do you see my screen? Okay. Before to start with a deep science subject, I want to talk about you about a little experience in my past. During my PhD and my postdoc, I met a lot of problems because I worked in an interdisciplinary team, so with experimental biologists, mathematicians, statisticians, and bioinformaticians. And I met a lot of limitations to produce high-level results. One of the limitations was collaborative space, a space to exchange results, documents, but also to discuss with my supervisor or other colleagues. It's one of limitation, but there are other limitations. For example, how I can share my results, share a report, share algorithm, or mathematical formulation. So during my PhD, I have this limitation. When I arrive in Gencovery, I discover Constellab, and Constellab, for me, is the solution to break this limitation. So as you can see on my screen, I built a little tabular to do a comparison between different tools, different existing tools. So you have Nextflow. So Nextflow, it's a software to build pipeline, especially for bioinformatics. We have Jupyter Notebook, RStudio, and Constellab, of course. And as you can see, Constellab have some strange compared to other software. For example, Constellab, inside the platform, we have a collaborative space. In this space, we can exchange with colleagues, we can build your project and manage your project, exchange file, exchange report results, and also share results with, for example, customer without limitation. In Constellab, we have also the possibility to run some code, low-level code or no-code level. This is one of strengths of Constellab. So for a biologist, he can use Constellab, but for expert in informatics, he can also use Constellab. So it's built for every people. So now I introduce Constellab and how does Constellab differentiate itself from traditional bioinformatics machine learning tools for omics data analysis. So I propose you to enter in a deep science. So like I said before, here this is the web page of Constellab platform. So we have different sections, section for project. So we can build your project with different hierarchy. We will see that after. And we have a section, we call that My Digital Lab. And what is a digital lab? It's a cloud, an access to the cloud to do computational exercise. So we have the opportunity to have a big lab or low-level lab. It depends on your need. So in this session, we will interest about data science and omics data analysis. So it's inside this project. So there are hierarchy in this project. So the first hierarchy called omics machine learning. Inside, I have a project for student project, and I have my project for this webinar. In this level, we can retrieve a section for report, a section for experiment run on the lab, and also a section for document. This section is, for example, to exchange a PDF or to write directly on Constellab some short report. For example, I will present the program of this webinar. So the idea of this webinar is to show you how we can use Constellab to build a pipeline to analyze multi-omics data analysis. So for that, I need data. So I take the data from one article, a very famous articles. So the idea is this data set is a multi-omic data set and is focused on a breast cancer. So we will detail the data set later, but this is a global context. So the pipeline. So there are 4 steps. So step 1 will be dedicated to the exploratory analysis. So descriptive statistics and visualization, associated visualization. The second step will be dedicated to the dimension reduction and data integration. In this webinar, I will not focus on data integration, but we can use some part of this pipeline to do data integration. So of course, maybe we already know there are a vertical integration or horizontal integration. In our case, we will focus on horizontal data integration. So for the third step is dedicated to the diagnostic and predictive analysis. So the idea of this step is build a machine learning model to train on the dataset and predict the behavior of the response variable. In our case, it will be variables, binary variables, so a survival variable, so 0 for people die due to the cancer or people survive. And the last step is the step of interpretation. We can this step is we can do multiple stuff. Generally, at this step, the biologists meet the statistician and try to find the good explanation of a statistical model. Okay, so let's go. So to present the pipeline, I come back on the first page and I will connect on my lab. But before I enter in my lab, I want to show you the parameter of my lab. We can see that here. So this lab is focused, it's based on Azure and is a basic lab. So with 4 core and 16 memory. Another nice option is Constellab. We can stop or start when you want your lab. So it's very useful to limit the cost, but also is very nice for ecology. So let's go. We enter in my lab. In this lab, we have different projects. This lab is connected with different projects, so I have many experiments. So in our case, we will focus on the project called Webinar Omics Data. And we can retrieve the different experiments linked to the different steps I explained before. So we will start with exploratory data analysis. So with this experiment. So this is the main page of the lab in Constellab. So this is the place where we build experiments. In these experiments, I have my data set here. So I upload my dataset in Databox, and I can retrieve, and I can import directly on my experiments with this option, add a resource, connected directly with Databox. Once we do that, we want to manipulate this dataset and explore this dataset. We have many options to do that. In Constellab, we have 3 different kinds of tasks. Task is like a piece of a pipeline. The first type of task is, we call that just task. It's already implemented task, so it's very easy to manipulate, especially for people with no knowledge in informatics. For example, here I import my dataset in a data frame, and for example, sorry, I want to describe my dataset with basic statistics. With this task called describe, we can calculate the statistic for each column of my dataset, and it produces results, a new table. And as you can see, in this table, we have the different statistics for each column. Maybe I forget to mention that, but in this dataset, it's a multi-omic dataset, so we have RNA-seq data, protein level data, mutation data, and count data. And this dataset is separated in 2 pieces, 1 piece for survival patient and 1 piece for died patient. So this is one of the type of the task. We have another type of task. It's called Python live task. Maybe you are already familiar with this kind of task because it's very similar to a Jupyter notebook. So if I enter inside, we can easily write a Python code to do manipulation of your input. For example, in this case, I want to describe my dataset. And for example, here I build a new data frame. And in this case, I count the number of variables for each type of omics data I have in my dataset. So for example, I have 604 features for RNA-seq and etc. The third task is called Conta environment live task. And it's a live task more dedicated to a bioinformatician. For example, if you want to import very specialized package to manipulate your data. Here is not very specialized, but for example, I need this package to do a plot, so we can easily import this package. And the last task we can have is a task linked with AI engine. And with this task, we can interrogate a NIAM engine to ask, for example, to produce visualization. It's very useful for people with no knowledge in code or for students. For example, if you want to learn how to code in Python, it's very useful to start with that. So now I describe the different tasks. We can explore the different type of results. So as you can see, we can have table. We can have also plot resource, so linked to Plotly package. So it's an interactive graph. For example, here I plot the distribution of a specialized gene, this gene in this case. And we have this task. And this task produces an image to plot a neat map with associated dendrogram. If I enter deeper in the subject, for example, with dendrogram, we can see clearly there are correlation between our different type of data. For example, here I plot the correlation between the RNA-seq feature and the protein feature. And we can clearly see there are correlation between certain gene and certain protein linked to breast cancer, of course. So this is the first experiment, and this is in this experiment, we explore the data, basically with statistics, and we try to visualize to have a better interpretation and better knowledge about dataset. So the next step, because this dataset is in high dimension, we want to reduce the dimension to obtain a nicer plot and to visualize better the different group in our dataset. So if I come back on experiment and not in training, in data science webinar, the second step is I call reduction dimension is this experiment. In this experiment, I start like previously with my original dataset, and I have two tasks. Here, I decide to propose you two solutions to reduce your dimension. The PCR solution, so a linear algorithm to project your data in 2D space, and another algorithm, more powerful, but this algorithm is not already integrated in Constellab. This is why we use a lab task to use this algorithm. So I suppose you already know this kind of algorithm. So the idea is reduce the dimension. So if I want to show the results, I click on the output of my my pipeline here, for example, and here I have my data set, my output data set. So the data in low dimensional space, in this case in low dimensional nonlinear space. And we want to plot these results. We have two possibilities in Constellab. We can create a lab task and use Plotly to plot the resource, but also we can directly, like in Excel, select column and create some graph. Here we have different type of graph. In this case, we are interested by the scatter plot. So this is the result of my projected data in a low dimensional space with UMAP algorithm. So as you can see, we can identify 2 or 3 clusters, one here, and one here. And for example, I want to superpose my response variables, so my vital status, die or survival. And because in this previous dataset, we tag each row with these response variables, we can easily superimpose these variables on my data. As we can see, there are some clusters more dedicated to die patient and cluster more dedicated to survival patient. For example, here, this cluster seems to more oriented for die patient. It's interesting. Maybe in this cluster, we can retrieve some gene, some protein, or some mutation linked to breast cancer, and maybe it can be useful to explore this cluster to identify some biomarker. This is the idea of the next step. On this graph, this is an interactive graph, so we can easily zoom in, for example, on a specific part of this graph. Okay. If I want to show the result with the PCA, clearly the results are very similar. but we can do the same operation, and we can visualize the result of PCA and superimpose the vital statue here. We are happy because there are no difference, no lot of difference between two algorithms. Of course, with UMAP, because it's taken in account the nonlinear part in the data, we are more separated clusters than in PCA. But like for UMAP, I can zoom in on specific parts of this graph. So this step is very interesting because when we explore the entire data set, we can see there are some combination. We don't know which combination, but some combination between features to maybe explain the survival status, so the possibility to survive or die due to the breast cancer. This is the first step, and the last step of exploratory pipeline. It's nice. So we have data, and it seems with this data we can explore deeper the relation between breast cancer and omics data analysis. So the next step is to build a machine learning algorithm model to explore the relationship between the different variables and how we can explain the survival status with this kind of relationship. So if I come back to experiment section, I have another experiment I call random forest, because I use this algorithm. Of course, we can use other type of algorithm. There are different algorithms to do that. For example, some algorithm linked to neural network. In this webinar, we don't have time to explore other algorithm. I focus on random forest because it's a very easily to understand and very powerful to manage large data set. So, like previously, I have my data set as input. I import this data set to transform this dataset file to data frame, and I create a Python lab task to build my model. If I enter rapidly in the code, very rapidly, we can retrieve one step to split my data. I want a training set, and I want to validate a test set or validate step. I want to build my model, I want to train my model, and I want to predict also my model on my test data set in this part. And in the second part, I want to explore my prediction. And for example, with random forest, a classical function to explore the output of the model is to run permutation importance task. I do that here. And because it's a machine learning algorithm, I want also to control how I train my model, if I train well or not. So with this task, as you can see, I have one input and I have multiple output. In this case, 4. So if I explore the results, we can start with a ROC curve, for example. This plot, as you can see, is show you how my model learn on the data. So the training is good, is not perfect, of course, but is enough good to continue the analysis. So on this graph is still interactive, and we can retrieve the training for the class 1 and the class 0, so for survival patient and die patient. Okay, nice. I train well my model. Now I want to explore the feature importance, and for that I create a plot to explore it. And on this plot, it's a very large plot, but if I can zoom in directly on it, I have all my features with different feature importance. And to explain deeper this kind of plot, more the feature importance is higher, better is it. But in this case, we have low value. It's normal. This low value is due to a high dimensional dataset. So we have many variables with no meaning behind. So of course, these variables bias the results. But this is the idea for random effect. Is not a lot influenced by this case. Okay, it's nice. I can extract the different feature, the importance of a different feature. I need to predict if my patient survive or die with breast cancer. So it's very useful. As we can see, we have some feature come from RNA-seq, some feature come from protein level, and maybe I have other possibility. For example, this feature come from a count number. So this is the first step to identify some biomarker linked to the severity of breast cancer. Of course, this graph is again interactive, so we can zoom in and zoom out as you want. I don't precise that, but we can also download this plot to put in a report here with this option. So you can download as you want the report or add in a report. We will see that after how to create a report in Constellab to explain the result and share the results. Okay, this is the main step in this webinar. So this is how we build a machine learning models to explore data analysis. Here, I do that with Python live task because I need some specific option. But of course, for example, if I want to, I don't know how to choose my option and I'm a beginner in machine learning, we have the possibility to have already implemented tasks. For example, if I choose that, here it's another algorithm to classify my data. And I have a task for prediction and a task for training. So if I click in, we retrieve a task and we have different type of parameter here. And we can directly link my table here and produce an output. Here, because it's a training, I want to connect directly with the predictor. And here I need the data. Oh, okay. As you can see, it's very easy to manipulate a pipeline because it's done graphically and is adapted for people with no knowledge in bioinformatics or statistics, or for experts, for example, with Python live task. Okay, we will finish on this kind of pipeline. Now we have a set of gene or set of protein, and this set of protein or this set of gene are potentially a biomarker for breast cancer. And now what I want to do, I want to explore the mechanism behind this subset I produce with random forest algorithm. So we can do a lot of things. Here, I focus on enrichment analysis, and I want to know if in this subset of gene, for example, I have some pathway enrichment. This is the idea of this step. This step, taking input a list of genes directly directly from the previous step I just explained, and I produce different image, and this different image is linked to different pathway enriched from the data, from the gene list. For example, if I explore it, I have different type of pathway. I'm not biologist, so it's difficult for me to interpret. But for example, if I search some stuff, we can retrieve some pathway directly link or indirectly link with breast cancer. Okay, it's nice. We build the pipeline. Now I want to create a report. And this report, I want to explain my results, and I want to connect the result obtained in experiments to produce a report. If I go here we have different type of report, but for example, this report is for this webinar. And we can retrieve the experiments I already present you. Is directly linked to the report. So if you change your experiment, the report will change also. It's very useful because you don't need to create a new report because you change the data or change your pipeline. In this report, we don't enter in the detail, but I present the method differently, and I want to show, for example, here we can retrieve the interactive plot I produce, and in this report is important, is still interactive. So, for example, for another user or a customer or a colleague, it can directly explore the results on the report, and it's not necessary to go inside the pipeline to check or to obtain an image. We can directly connect the output of the experiment here. As you can see, we retrieve the different output of my pipeline. For example, the PCR results, the UMAP results, etc. The red curve, the feature importance, and also different pathway. For example, here I plot the estrogen signaling pathway directly linked to breast cancer. And when you can see in red is the gene I identify in my output of roadmap forest. So it's a potential biomarker to explore if the patient died due to the breast cancer or if it survived, and maybe it can be useful to drug discovery or other next application. Okay, I finished the part on the science. Now I just want to show you a nice new feature in Constellab. As you can see, I present you different type of tasks, and I don't take a lot of time of this task, but this task can be shared with other people, with your colleagues, but also with other people as you want in Constellab. And for that, we have a website called Community. Here, I already opened this page, and on this web page, we can retrieve a section for story. This section is dedicated for use case, for example, for build metabolic pathway and reconstruct and analyze this pathway for metagenomics, etc. We have the documentation, the low documentation and high-level documentation, the different bricks we have in Constellab. So I present here the brick linked to Gaia, so machine learning algorithm, but of course we have other brick for modelization and specialized for the single cell RNA-seq data analysis or other stuff. And we have a last section, last task section, and here it's a space to share some part of your pipeline. For example, here I upload a last task to produce the UMAP algorithm output. It's very useful because if I come back on Constellab and on my lab, for example, I just show you one example. But here, if I want to retrieve this task, of course, we can modify with some knowledge in coding, but also we can directly add a community lab task. If I click here, I retrieve all the lab tasks upload on community website. So it can be useful, for example, if I want to plot a curve, if I want to visualize pathway, for example, cake pathway, etc. So this kind of lab task is the intermediary between classic lab task like Jupyter Notebook and already existing implemented task like, for example, this task. I think I finished. I just come back on the space to show you on this page. As you can see, all experiments report are directly connected to the project. So here we retrieve the report, and here I retrieve my different experiments. And we have also nice option. We can communicate with colleagues or members of the project directly with a chat.